Our next speaker is Simon Bekker-Jensen from the Center for Healthy Aging here in Copenhagen.
Thank you, Daniela, and thank you to the rest of the organizers for inviting me here. So I'm interested in, it's coming up, right? Yes, there it is. So I'm interested in cellular stress responses. And the biochemical systems that I'm really focusing on is MAP kinase signaling, especially by those two kinases up here, P38 and JNK, that we also call the stress-activated protein kinases. We also have the ERK signaling pathway, but that is quite different from these two. Now, upstream, it's important to know that downstream of P38, there's a lot of downstream effectors and a lot of phosphorylation targets, and even P38 has tons of downstream kinases that mediate hundreds of phosphorylation reactions. Upstream of these MAP kinases, there is a whole cloud of so-called MAP3 kinases, which are really the ones that sense the different signals coming from inside or outside the cell and mediate the activation of the downstream stress signaling. Interestingly, this cloud of MAP3 kinases is much more vast than the other kinases, and it's really very poorly understood how they are activated and what kind of physiological roles that they play. Now, when I initially started my lab, I had a background in DNA damage response signaling, and then when I started my own lab, I thought, let's take a leap, and study all the other things that cells don't like, so all the other types of cell stress. And these are things such as osmotic stress, it's a ribotoxic stress that we call it when translation is impaired, it's oxidative stress that gives a lot of ROS from metabolism, it is changes in temperature, and it's mechanical perturbations to cells and tissues. And I was really delighted to see that I was going after Suresh, so that you're all primed for this kind of thinking. When I tell people at a dinner party that I study stress, they invariably think about this, that we all know stress is a very, very bad thing, but it's also, of course, known that we need some kind of challenges to actually perform to our best abilities. No stress, no activity is also a very bad thing to our body. And actually, there are very similar conditions for our cells. It's also well known that if we give a lot of stress to individual cells, well, that can result in cell death, senescence, even transformation of cells. But on the other hand, those small physiological stimuli that all our cells receive all the time, which are actually small stress stimuli, they are also key to mediating or to triggering adaptation of our organs, and really are key to a lot of the physiological responses that our body has to have. The ability to all the time adapt to small physiological input and small stress stimuli is really a key for healthy aging. Now, with all that said, the activities in my lab can really be boiled to this small graph here. We want to understand what the molecular signals of these stresses are. What is it actually that the cells kind of recognize as a stress stimulus? How is that signal sensed by MAP3 kinases? And finally, how does the downstream signaling from these initial signals promote organismal fitness and longevity in various organs and systems? And I have two small case stories that I want to highlight here. Now, the first thing that we actually started studying was something called the ribotoxic stress response. And that's actually, it was kind of well known that if you do something and inhibits translation in the ribosome, well, that leads to very strong JNK and P38 activation, and this can even kill the cell. And classic ribotoxic stressors were things like ricin toxins, it was inhibitors of the ribosome such as cyclohexamide, and UV radiation was able to elicit this signal as well. And here we had a clear idea that the signal must somehow originate at the ribosome, and it was also shown that there's only a single MAP3 kinase that can kind of detect these signals, and that one is called SAC. So we started to study the SAC gene. Turns out that in mammalian systems, there is actually two isoforms of SAC being produced from the same gene, and they have two different, they're splice variants of each other, but in such a way that they have unique C-termini. So that this beta isoform is not just a truncation of the alpha isoform. we were able to map two ribosome binding domains on the long isoform Seg-alpha and could show that these two interact with the ribosomes to read the signals of translational impairment. And this is a response that is mediated exclusively by Seg-alpha and not its shorter isoform Segbeta. However, it was also clear to us from looking at expression data that the longer isoform Segbeta is often something like 10 times higher expressed than Seg-alpha in different tissues, and in all the cell culture models we studied, there's a lot more Segbeta than Seg-alpha. But what this one does, it was completely unknown to us. So what is then actually the molecular signal for this kind of Seg activation? So that has been shown by others as well as by us, that it seems that a general feature of detecting translational stress is to detect collided ribosomes. So whenever one ribosome stalls or is impaired, it will be caught up with by the trailing ribosomes, and these ribosomes will collide, and this is a kind of molecular trigger of many of the downstream pathways, including this p38 activation mediated by Seg-alpha. I see this as what I call an orphan pathway, in that we now more or less understand the mechanistic basis of how ribosome inactivation leads to p38 and junk signaling, but the only way that it has been studied so far is in human or animal cells, using things like UVB, ricin, cyclohexamide, all completely unphysiological agents. So what is really the physiological relevant triggers of this pathway? Why is this kinase Seg-alpha even conserved evolutionarily? And in some of our recent unpublished work, we have shown that amino acid starvation is actually one of the things that can trigger this. And that makes a direct link from starvation to metabolic regulation via the p38 and junk signaling pathways. Now, we study these kind of responses in relevant organisms, C. elegans, the mouse, and the long-lived zebrafish, where we can study, both aging, longevity, and metabolic regulation, for example. And as I said, this is just a little teaser. So if we take our cells that are depleted or normal cells, and we starve them with starvation medium, we get very strong p38 activation, and that is Seg dependent. If we then take our Seg knockout worms, we can see that if we compare the overall lifespan of completely unstressed worms that are either deficient for Seg or p38, this doesn't really rock the boat. They have more or less the same lifespan. But whatever we do to stress these worms just a little bit, for example, by starvation, then they become very sensitive. And by whatever, I mean, if we also, for example, increase the temperature a little bit, then suddenly we start to see lifespan defects of these worms that cannot mount this ribotoxic stress response. So this is probably the inability to respond to low levels of stress that gives these phenotypes. What I really wanted to talk about today was the other MAP3 kinase encoded by the same locus, namely this Segbeta. As I said, it has a unique C terminus, so it's not just a truncated version of Seg-alpha, and it turns out that this kinase is activated when we apply osmotic stress to cells. It's done here by sorbitol and sodium chloride, and that gives this huge kind of mobility shift of the Segbeta kinase that we showed was due to autophosphorylation. We could also show that Seg deficient cells were kind of deficient in the ability to activate p38 upon sorbitol treatment. It is not a complete defect, but that is because there are other kind of osmosensitive MAP3 kinases in the cell. Now, sorbitol, when you apply that to cells, it's kind of, it's an osmotic stress, but it's also kind of a compressive load that you apply to the cells. And we think that this is actually what the cells are responding to. So it's a mechanical perturbation. Now, we found out that here we have GFP Segbeta expressing cells, and you see that it's largely in the cytoplasm, a little bit of it in the nucleus as well. But when we apply sorbitol to these cells, the protein is extremely rapidly kind of recruited to cytoskeletal like structures, and we thought that this is kind of the initial signaling event. Similar to how seg-alpha recognizes ribosome, seg-beta seems to recognize something being perturbed at the cytoskeleton. And we can see that these fibers were actually what we call stress fibers. They co-localize completely with alpha-actinin and also kind of end up in these focal adhesions marked by, for example, the focal adhesion kinase. I hope you can see some of the details. Now we wanted to kind of understand where that property comes from, so we made different truncations of the Segbeta construct, and for example you can see the wild type here is very rapidly recruited to these stress fibers, but if we delete the C-terminus that we don't find in seg-alpha, we lose that property. And kind of very convincingly, if we just look at the very last fragment of the protein and hook that up with GFP, that one functions as a sensor itself and will go to these stress fibers in the presence of this compressive load. Importantly, if we try to rescue Sag-deficient cells with this truncation, we see that we can no longer rescue activation of p38 fully by sorbitol. So this sensing reaction seems to be connected to the activation of the kinase. Now, this turned into a mechanobiology project because we understood that it is kind of a mechanical perturbation of the cells that we're looking at. And to make a long story short, we found out that this kinase is sensitive to compressive load. So if we compress cells, for example, here embedded in an agar matrix, well, then we get p38 activation, and that is completely Sag-dependent. Importantly, if we stretch the cells, we also get p38 activation, but this perturbation is not Sag-dependent. So there's different mechanisms for compressive loads and for stretching. And importantly, again, here we see that this property of Segbeta to get activated by compressive load, again, requires its ability to kind of interact with these stress fibers. So we think that is a good case of a signal originating at stress fibers that is being sensed by a sensor domain in this Segbeta kinase. So what is this mechanism then good for in a physiological setting? Now, as I said, there are two isoforms of Segbeta and Seg-alpha, and if you look at the expression levels of them, very sensitive, you see that the upper one is Segbeta, and across tissues it is much higher expressed in general than Seg-alpha. And it turns out that there are actually Sag knockout patients out there who have different mutations, stop mutations, for example, right in the kinase domain, and must therefore be considered kind of null mutants. Now, when that was published, I was very surprised to see that these patients actually suffer from a congenital myopathy. So they have decreased strength of their muscle, hypotonia. But when I then looked at the skeletal muscle expression, I could see that this is the organ with the highest expression of Segbeta and actually no expression of Seg-alpha. So if we look in that organ, we can kind of surmise that the defects that arise there are due to the lack of Segbeta function and not kind of because there's no Seg-alpha. So of course this led us to speculate that maybe the compressive load associated with muscle contraction is what is kind of one of the physiological signals to activating this pathway. And we looked at that, not in humans, but in a mouse, of course. And the way you do that is like this. So you anesthetize a mouse, and then you put this little electrode into the thigh and stimulate it to actually doing physical work. This is called in situ contraction. And for muscle physiologists, this is, of course, everyday business. But for people like me who are not in that field, this is quite fantastic to see, right? So you do that for 10 minutes, then you kill the mouse, take out the muscles, and look, for example, by Western blotting at the activation state of different pathways. And as you can see, while contraction is normally associated with an upregulation of p38 activation, this is more or less completely blunted in the knockout mice. Really kind of saying that, this is something that this MAP3 kinase Segbeta responds to, and this is an organ where it's physiologically relevant. And we see that other markers of exercise, such as AMPK phosphorylation, is not altered. That's kind of a control for that they were exercised indeed. So where does this kinase localize in the muscle then? Well, it turns out that it also in this organ has a precise co-localization with alpha-actinin, which you find at these Z discs. So Z disc is kind of the muscle equivalent of the stress fiber, and where it is believed that force generation is kind of being sensed in the muscle fiber. But there are clear kind of correlations between how this all works in specialized cells like muscle fibers compared to what we see in our normal cell cultures. Whoops. And how are these mice doing then? Well, it turns out that just like the humans, Seg knockout mice actually have a very clear muscle pathology. So I've just have a single data slide on that here. And the sign of that is that you see these centralized nuclei in the muscle fibers in Seg knockout mice. This is something that is stronger in males than in females, and it's also something that is kind of increased with age. These are still young animals, but we think it's not a congenital kind of defect, but something that arises due to the use of the muscle, even though we haven't really proven that yet. It's also quite striking that there's a huge difference in the degree of pathology between mice and humans. So humans are really handicapped from this mutation, and they actually sit in wheelchairs. We're talking about small children who never learn how to walk, for example, but these mice overall look completely fine. And it's interesting that the only muscle where we see a very clear pathology like this is actually in the soleus, which is the best example of a slow twitch muscle in the mice. So there's probably something with the difference in muscle in muscles between humans and mice, where most of our muscles are very much like intermediate in their kind of distribution between type 1 and type 2 fibers, whereas most of the muscles in mice are of the fast twitch mice. So we are maybe built for running, and mice are built for jumping. So that's probably the reason why these animals are still doing quite well. So now we are kind of making further interrogations into the physiological importance of this signaling pathway. So we are looking at, for example, these mice, whether they can produce myokines upon exercise, whether they can, repair the muscles after injury, can they make, improvement in their physical shape with exercise, etc. But I think it's a very nice case of a stress stimuli, which is actually turning into something good in an organism. With that, I just want to thank my collaborators. This is an ancient picture of my lab. Thank you very much for the time.
Thank you, Simon. We have several questions on Slack. So the most upvoted question is from José Navarro Betancourt. He's asking, rapamycin inhibits protein synthesis. Does it also induce ribotoxic stress?
Yes, it's actually very recent data from our lab that so ribotoxic stress actually seems to be boiled down to collision events, and it turns out that inhibition of mTOR actually induces collisions. We have shown that now in many biochemical ways, but it's unpublished. It's not kind of known. We don't use rapamycin, we use taurine instead. But that leads to collisions and upregulates Seg activation, for example.
Okay, maybe we have time for one more question from Wilbert Vermey. Is there a common molecular or signaling denominator in all these different stressors?
Yeah, so So downstream, you get P38 and JNK activation after all of them. What I find fascinating and very hard to wrap my head around is that you have so many pathways leading from many different stimuli to the same signaling output, and it has so different consequences based on the cell, the tissue, the duration, and so on. Right? How can you, with the same input signal, get so diverse outcomes? I don't know if that was a question, but I think that's a thing that we really need to understand better in the MAP kinase signaling field.
Okay, thank you very much, and maybe you can give another applause to Simon and